Hello and welcome to this webcast, Strategies to Revolutionize Research with Seamless Data Access for Scientists, hosted by Scientific Computing World and sponsored by Sapio Sciences. My name is Robert Rowe. I'm the editor of Scientific Computing World, and our guest speaker today is Rob Brown. Rob Brown is a senior director of product marketing at Sapio Sciences, and is a product management executive with more than 25 years of experience in life sciences software and pharmaceutical research. For most research organizations, having all their scientific data organized in a way that scientists can easily search, visualize, and analyze is a key milestone on their ongoing research journey. However, many organizations see data consolidation as a destination, rather than just a step on this path. But on its own, consolidation without scientific context doesn't help scientists, forcing them to go through too many hurdles to make use of this data. So what's the solution? In this 30-minute webinar, Rob Brown will take a closer look at laboratory data strategies that put our scientists and research first. After the presentation, there will be a question and answer session, which anyone can use to ask our speakers a question. If the speakers do not have time to answer your question today, the information will be passed along to the relevant people so they can follow up at a later date. Rob, over to you. The floor is yours.
Thank you very much. Good morning, good afternoon, everybody. Let me just share my slides and we will get started. Just confirm you can see the slides okay?
Yep,
okay.
Perfect.
So again, thank you very much for the introduction. I'm going to start with a quick introduction to Sapio Sciences for those of you who are not so familiar with us. Then I'm going to talk about the data problem that you just heard about in the introduction and how Sapio can help you with a solution. And then I'm going to finish up with some work that we've just released, adding in AI assistance on top of that data solution. So for those who are not familiar with Sapio, we are a research and clinical informatics company. We provide an ELN, a LIMS, and a science cloud called Jarvis, which is the topic of the main part of my talk. But these are all seamlessly integrated with each other because they're all built on a common platform and they all share a common set of data objects. The platform is completely low code, no code, so it's very easy to configure any workflow that you have across the platform. But to allow everybody to get started quickly and up and running, we also provide a set of out-of-the-box pre-configured workflows spanning research and clinical, from medicinal chemistry, next-gen sequencing, histopathology, all the way through to bioprocessing and stability. And as I say, underpinning all of these is a common platform with a set of shared services. And you're going to see many of these in action today. Everything, as I said, is low code, no code, including the ability to fully configure the data model, to be able to configure rules that trigger on particular events in the software, and also no code querying without the need to provide any SQL. We have very advanced charting, visualization, and analytics that you'll see as we go through the Jarvis capability today. And then towards the end of my talk, as I say, I will come to the AI and chat components. And again, because they're in the platform, although you'll see them primarily in Jarvis, these would equally apply if you were using the ELN and LIMS. So with that quick introduction, let's get to the bulk of the talk. The starting point for this is that research still has a very big data problem. And don't take my word for it. This is a really nice survey that was done by the Pistoia Alliance at the end of last year. They polled over 200 pharma biotech research organizations. One of the questions was, and this was in relation to scientists consuming data, what are the biggest barriers to making use of scientific data? And over 2/3 of responding companies said that the biggest problem was data silos and accessibility. And that's the problem I'll come to in the next section of the talk. Just below that, 58% also said accessing unstructured data was a really big problem. I'll try and address that as we go through the AI section. And then interestingly, third, resistance to data sharing was still called out as a problem as well. Very importantly for thinking about how we go forward in the future if scientists still have a data problem, that's going to mean that AI still has a data problem, because the most fundamental thing that AI needs to be able to build models to assist scientists is really good data to drive it. In that same survey, when questioned what are the biggest barriers to implementing AI ML at scale within the laboratory environment, over half called out low quality, poorly curated data sets, and then followed up by data that is not fair. So again, as we go through the talk, hopefully you'll see how Jarvis can begin to help you not just address the science problem, but also the data preparation for AI problem. So if we just come back to that diagram I started with, if we lived in an ideal world, and here I'm defining ideal world very narrowly from the point of view of a Sapio sales manager, in an ideal world, if in every company you only had Sapio ELN and LIMS for all of your workflows, then actually you wouldn't have a data problem. Because everything's built on a common data core and all of those common data objects are shared, all the data is organized, it's easy to access, and then with the platform services for search and visualization and analytics, all the data is easy to find, to visualize and analyze. But of course, that ideal world really doesn't exist very often. And for those of you in companies of any scale, any history, you'll know that the reality is much more what you see on this slide, which is it is very common to have different ELNs, different LIMS, different informatics systems across different departments, different scientific domains, different systems because two companies were acquired and came together, so different histories essentially. The real problem with this, of course, is this is the place in which all those critical scientific objects relevant to a scientist understanding their problem live. So within these systems are the samples and specimens and the experiments and the workflows and the projects that those were part of. And then complicating this landscape even further, the more the industry moves towards automation to actually conduct the experiments, then there are a whole fleet of instruments. And of course, those instruments are generating results, but those are results pertinent to those samples and specimens in those informatics systems. So we kind of all know what happens over time, which is over time, point connections get built up between the individual systems and the instruments, and actually even between the individual informatics systems themselves. So this is a problem, firstly, because not just building, but maintaining those connections is costly and time consuming, but much worse from the point of view of those scientific data problems that I talked about in the survey. Your most valuable asset, which is the data, is siloed, it's not harmonized. And frankly, if I'm a scientist coming into a landscape like this, trying to find everything I need to know about, let's say, the samples in my project, it's almost impossible for me to be sure that I can gather up all that information. Now, I do understand this is kind of one end of a scale. There is very definitely a transition towards scientific data cloud-like solutions across the industry to try and address this problem. But honestly, what we're seeing in a number of scenarios is that customers are actually just layering in data lake-like solutions on top of this. And I say solutions advisedly. You sometimes see more than one of these. This can absolutely help cut down the number of connections between the source systems and to the instruments. But this landscape is no better than the previous one when you think about scientists finding all their data. The data is still incredibly hard to find. The data lacks scientific context, and I'll come back to that as an important point in a minute. So that makes it very hard to search and navigate. And frankly, if you look at a lot of these data lake solutions, they are designed for IT and data scientists. So scientists can't or won't use them. They fundamentally lack scientific awareness, which is the key to understanding the scientific data. So this is where Jarvis comes in. Hopefully most of you get the reference to the Iron Man movies. Jarvis was a digital assistant, short for just a rather very intelligent system. And what would be ideal to address those two scenarios I just showed you would be a rather very intelligent science system and not just another data lake. So if I'm going to claim Jarvis is intelligent, I'd better actually set out to prove to you that it is. I will try and do that over the next few slides, but here are kind of signposting the things that we think make it scientifically intelligent. As you'll see as I go through, as data comes into Jarvis, it's contextualized. That gives it meaning. That's what fundamentally allows scientists to navigate and search the data. Jarvis has complete scientific awareness. That's what gives scientists the ability to visualize and analyze scientific data and not just text and numeric data. And then finally, science is always evolving. So within Jarvis, thanks to the no-code platform, it's incredibly simple to extend the data model to encompass types of data or attributes of data types as those are essentially created in the scientific teams. So let's take a look at a high-level data flow, and then I'll drill down through the various parts. So the premise is, as I said, on the one hand, you have a set of ELNs and LIMS across your company, and within those ELNs and LIMS are references to samples and specimens, the scientific entities, small molecules, large molecules, etc., that they are samples of, the experiments they were produced in, the LIMS workflows they've been processed in, maybe the projects they even came from. And then on the other hand, you have a whole fleet of instruments. And the instruments might be writing files to connected desktop PCs. They might be writing files to network drives. If we're lucky, the instrument might come with some software API that can actually communicate the raw instrument result over the API. And then the final source of data is that you likely have a set of document repositories, coming back to that problem with unstructured data. I'm going to set the documents aside for the last section of this talk and really just focus in on the flow between the ELN, LIMS, and instruments for now. But essentially what Jarvis does on a schedule is collects up all of that information. Now, really importantly, it's not synchronizing every piece of data from the ELN and LIMS. It's synchronizing the important pieces of data that it later needs to understand the instrument and test results. Because the next step, having just collected everything, the critical step is to contextualize that data. So just to give you a really simple example of what I mean by that, imagine in an ELN, we have an experiment with a set of samples. Scientist goes off to an instrument, runs those samples, and once we've parsed and understood the instrument output, then we have a set of measured results relevant to those samples. We could even then go into a piece of software and calculate some more derived results from those measured results. What contextualized means is put the calculated result in the context of the measured result, in the context of the sample, in the context of the experiment, in the context of the project. So essentially connect all of that data up and make it discoverable. And again, do this across the multiple systems that you have in-house. Having done that, we can do two things. The first is we can automatically then insert the result back into the experiment or LIMS workflow that it came from automatically without the scientist having to collect up files, parse them, manually upload them, and put them in their experiment. But more importantly, as long as we have a system that is user-friendly for scientists, this is really now the place that the scientists can go in. They can explore their data based on navigating the knowledge graph or searching it. They can apply analytics to discover new information. And increasingly in the AI ML world, they can actually trigger the creation of new analytic models that further help them understand their data. And with all of that, move on with their project. So if you look at that circle, the left-hand side and the top are essentially the data automation piece that all happens behind the scenes. And then the right side and the bottom are the scientist experience where they actually interact with the results. So let me go through the top and the left, the automation piece first, and then we'll come back to the scientist experience. So here's a slightly different view of the first part of that diagram, just to drill into the data automation. So on the one hand, we have a set of ELNs and LIMS. I deliberately did not put the Sapio logo on here. There is absolutely no requirement that any of the source transactional systems be a Sapio system. But again, what happens on a schedule using those applications APIs is that we synchronize across not all of the data, but the data that we will later need to contextualize the results. So samples, specimens, experiments, etc. And then on the instruments, we're watching and listening for new files to be created with file watchers, or communicating with the API, waiting for new results to occur, uploading those to Jarvis and parsing them to produce structured data in a way in which I'll talk about in a minute. The real key then is the data processing, and this is all managed with the no-code rules engine. So within the software, you could create a rule such as the example I've showed you there. Imagine we have a BioVia ELN, which has an experiment with a set of samples in it, and then scientists has run those samples on a SciEx, and back have come a set of results. The rule says when the raw SciEx file is detected, add the result by syncing back to BioVia. What actually has to happen behind the scenes is a couple of things. First is connecting the data to the proper context inside the knowledge graph, which I'll show you on the next slide, and then using, in this case, the BioVia API to insert those instrument results back under the sample in the right experiment. So what does that look like in practice? Well, imagine this is whichever ELN or LIMS you are using, and within that, you've gone in there, you've created an experiment with 2 brand new samples. You have, as I say, then gone into the lab and you've processed those samples, and out has come an instrument file. So the first thing Jarvis has done is collected that file, sent it off to the parsing service, and then back has come a set of structured results. Now, behind the scenes, on a synchronization schedule, as soon as that ELN experiment was created, the Jarvis knowledge graph has automatically been updated to include that experiment and those 2 samples under my project, such that when Jarvis then produces or has determined results, it's not just saving the results under the file, it's actually inserting them in context under the relevant samples. So you see under the 2 samples on the right-hand side, now 2 SciEx results. Now, I know I keep saying graph and showing you tree. I will come back to that later. The tree is just a nice representation of a graph at a very high level. The final step in this, as I said then, is using the API for whichever ELN or LIMS you're using then. Now we know the context of the results, we can insert them back into the right experiment, linked to the right samples. And again, without the user having to do any manual processing at all. So obviously one of the keys to the whole data automation piece is how do you parse the instrument files? Because we really need to understand all the kinds of instruments you may have. For that, we are using a service called LDAS. And the way that this works is that LDAS is already built with over 200 parsers for common instrument files. This is built and maintained by our good friends at Ziffo, and they have the ability and the team to develop new parsers very rapidly as other instruments coming up. They're always expanding the library of parsers. They have built a Jarvis agent inside their LDAS service such that when we receive a raw instrument file, send it to LDAS, LDAS does its thing, parses the result, pushes us back the structured data. The workflow you see on the right-hand side there is specifically if we're receiving a file and sending it. The same infrastructure with a slightly different workflow would be used if we were connecting to instrument APIs. So if you have Empower, Chameleon, OSI Pi, those kind of things, that's how we bring the data across and structure it then. So all of that, if you remember that cycle, was the left-hand side and the top. We have now got the data in, we've contextualized it, and we've built the knowledge graph all completely automatically behind the scenes. The next step then is, this is a resource now that a scientist can use, again, provided you build the correct user interfaces that really let a scientist interact with the data. So this is what a scientist would see as they log in. On the left-hand side is the top-level tree representation of the knowledge graph. And hopefully you can see what we've got in this example is we've got 2 or 3 different instances of the Sapio ELN and LIMS hooked up to represent different kinds of software in different departments, but also a benchling instance and a Dotmatics instance. And the user can simply open up and explore down in one of those Sapio instances. I can see my projects and my experiments and my samples. And then below that, we see all the connected instruments. And if I were to open up one of those instruments, we'd see all the files that have been synchronized in. So the scientist immediately has a place they can go where all the data is collected across all of those transactional systems without them having to know how to get to each one or how to find the data. As I say, it's very easy to add new data types, so anything can be added into this tree by defining a new data type and its attributes in the admin capabilities. And then using the visualization within the software, dashboards can be built very quickly, again, with no code. So an example just at the very top level shows the number of connected instruments, the number of connected applications at the bottom, and then some counts of the total number of objects that have been synchronized across into the system. So just to come back to the point that I keep showing you trees and saying graph, it really is a graph underneath. And at some point, as you zoom, as you walk down through the tree, you actually get to the point where you really can understand a chunk of it in graph representation. So if I were to walk the tree down to a sample and then drill through, this is the kind of thing I might see. So in the middle here, slightly light blue, we see a DNA sample. And what I can see if I walk back up the graph is that DNA sample was extracted from a blood sample as part of an experiment that you see at the top there. And then what happened to that sample is it was then aliquoted into a DNA library, and then it was pooled with a whole set of other samples. And then presumably later it went off to an instrument to be QC'd or to be sequenced, and then we would have instrument results coming in under the pool that could then work their way back up to that sample. So there's a lot that can be achieved by walking the graph and navigating. At some point, you obviously have so much data that you really need to search to very quickly find the thing that you're looking for. The Jarvis inherits the platform's no-code search. So this kind of search applies all the way across the software. It is very easy for an end user to simply set up searches. In this screenshot, it says, find me all the samples where there's with a SciEx result with a calculated concentration greater than 400, and it will return the table of data with some drill down links and some visualizations of the data as well. So text and numeric search is very easily built up using this no-code query builder. And then if we want to ask questions of scientific objects, substructure search for molecules, blast search for sequences. So that's a very important point, and that's what really makes this begin to stand out from data lake solutions, is I can ask a scientific question like substructure search or blast search. Equally importantly, I can then also visualize the results, or in fact, any object in the knowledge graph that's a scientific object in a form that makes sense to the scientist. So if, for example, I'd hooked up a set of large and small molecule registries into as one of the data sources, or there were these objects within the ELN experiments that were being synchronized across, I can immediately visualize in this, on the left-hand side, a protein with a crystal structure. If you think about the alternative would be, having found all that information, I would then have to download a PDB file, fire up PyMol or my favorite viewer, load the PDB file, and then visualize it. So we're bringing all the scientific visualization right next to the data where the user already is, which makes this a much more seamless experience. Same thing would apply if there were annotated sequences, as you see on the top right, or if we were looking at small molecules on the bottom right. And then to take that one step further, it's not just about the visualization, it's also about the analytics. So anytime I have a table of data that I've brought in from my multiple source systems, I can immediately fire up an analytics wizard, and in place, without any file transfers, I could do ANOVAs or dose responses or t-tests, as you see at the top left, producing the kind of output you see at the top right. And again, the alternative would be, having found my data, download a file, fire up Excel, MATLAB, GraphPad, Prism, whatever my favorite analytics statistical engine is, and then do my result and bring that back where I came from. That applies not just to statistical analysis, but also to scientific analysis. So imagine Jarvis was synchronizing in was connected to a whole bunch of flow cytometers, and it was automatically syncing in the flow cytometry output and then parsing it automatically to pull all the channel information out. In place in Jarvis, as you see on the bottom right, I can immediately fire up a full flow cytometry analysis capability, in this case, do my gating, and then move on with my experiment. And again, didn't have to download files, fire up FlowJo, FCS Express, do my analysis, save the result, move back. And then the final example, which really shows the power of this, is on the bottom left, because it's often the case that you need to do analytics across multiple sources of data. So this analysis is of a Sartorius Amber bioreactor array. It's the 2x12 array, the small one when you do the workup and figure out the ideal conditions. But if you think about the outputs, there's really a lot of things happening, because sensor data is flowing off all of those vessels in real time, going into probably a historian where it becomes available. But then every time period, every day, for example, you're stopping and you're taking samples and you're measuring the number of viable cells and then glucose levels and all those kind of things. So to be able to analyze that data without all of the data automation, there's a huge amount of data collection and aggregation that has to happen. In Jarvis, we can simply reduce this down to the user to say, which sample and batch do you care about? Okay, that was processed in these vessels in the array. Which vessels do you care about? What sensor data do you want to see? What sampling do you want to see? And put it all together automatically. So very, very quickly, I could take a look at this and say, if I look at the orange bars, which are the number of viable cells in the first vessel, clearly that was all going well until the middle of the run when it wasn't. And if I look at the sensor data, something clearly spiked in the O2 or CO2 levels. Whereas if I look at the blue one, vessel 2, that was a very nice progression of viable cell growth. So clearly condition 2 was better than condition 1, and I arrived at that very quickly. So hopefully what you've seen from Jarvis then is it's a really nice way to really begin to solve the data problem for scientists. And it also, of course, has huge potential to collect up data now in a well-curated way. That is exactly what AI needs. So Jarvis is scientifically aware in the sense that when scientists learn to use the software, it has kind of that scientific awareness. But if you think about it, it's somewhat passive in that the scientist has to learn how to use the software. The next step on the journey is, can we actually make the software scientifically intelligent so that it actually becomes not a piece of software for a scientist, but actually begins to more and more resemble a scientific assistant? So I'm going to give you 3 examples of that, again, all available in Jarvis and also all the way across the ELN and LIMS. The first one is Elaine. And the idea of Elaine is, how do we take the new paradigm that large language models, things like ChatGPT and Llama, are using to fundamentally change the way in which humans interact with software and large volumes of data. So the paradigm has changed, for example, from doing a Google search of the internet to going to ChatGPT and literally having a conversation where ChatGPT knows the entire contents of the internet and can refine information for you. So we're leveraging that in an assistant called Elaine, short for ELN AI. And that's how do we change the interface to the software to be more natural language. And then we're also leveraging it to actually search the contents of the informatics systems, including Jarvis, using that same paradigm. Those are both really revolutionary because they're fundamentally changing the software from being science aware to actually beginning to be scientifically intelligent. We've also used large language models to actually build a support assistant to provide more efficiency, and I'll get into those. But let's start with Elaine. And I'm actually going to go back to that search I showed you in the Jarvis section. So the actual problem statement in these kinds of scientific searches is very straightforward. In natural language, I want to find all the samples in my company where somebody's run a SciAx and the results come back with a calculated concentration greater than 400. Simple question. I just want the simple table of data that you saw before. Samples, calculated concentrations, filter by the right row, give me a drill down link to get to my result. But think about what you would have to do in other solutions or in these data lakes. Somebody somewhere is probably going to either have to write a SQL snip, it's like sample star from sample, or they're going to have to write in a data lake maybe a Python snip to be able to pull that out. Quick and easy for a scientist? Absolutely not. So as you already saw, the Sapio platform had already eliminated all of that even before we get to the AI assistant, and all a scientist had to do was to learn to break that query down into two terms and then graphically build up the sample term and the SciAx term with the calculated concentration restriction on it. But how much easier would it be to not have to learn to use the software at all? Just to get a prompt, either a text prompt to type or even a voice prompt. And then as you can see, hey, Elaine, find me all the samples where there's a SciAx result and the calculated concentration is greater than 400. And she'll go off and think, thank you for your request. Here you go, you've been redirected to the search. And what comes back is exactly the query that you saw before. So here's my result that I asked for. But also what Elaine does is show her work, partly so that you have reassurance you're getting the right result, and partly also as an educational tool. So I didn't have to learn to use the software. We've basically trained the AI assistant to learn how to do a search so that the user doesn't have to. But if they do want to learn, here's an example, and they could use this to refine the search. I'm not going to get into a huge discussion of how this works. The real key thing, as I keep saying, is we've actually had to train the large language model how to use the software in a way that the scientist would use it. One of the really key things then is understanding what the scientist means by a natural language request. So think about a slightly more complex prompt than the one I showed you. Find me all the samples I've created in the last month that have a SciAx result, et cetera, et cetera. What does the software actually have to do to be able to do the right search in the first place? Well, the first thing it has to figure out is, I said, find me. What does that mean? Okay, well, the user wants to do a search. So let's create a blank search. Find me all the samples. What samples? Samples are data type in the system. So Elaine needs to add a term to the search for sample. I have created, what's I? Well, I is the current user. The current user is R Brown. So Elaine needs to constrain that by created equals R Brown. Created in the last month. Okay, that's another constraint. It's a date constraint, and there's a macro for last month. Oh, and by the way, I have created has an implicit and in it. So we need to and those terms together. That have a SciAx result. What's that? That's another data type in the system. So that means we've just created another implicit and. What's calculated concentration? It's an attribute. What's greater than 400? It's a constraint. So a lot of work has to go through, because I could have phrased that question in any number of ways to get me the right search, to get me the right answer. But again, having done it, I've now completely relieved the scientist of doing mundane things in the software and I've freed up their time to go just do something more productive by actually looking at the search result immediately. So that's using large language models to change the interface. How about using large language models to actually process the data within the system? If you remember back to the beginning and that survey, one of the key things that was mentioned in the survey is the problem of accessing unstructured data. And this is because many of you, any of you in a company of any size or any history, will have a huge legacy of document collections, research reports, old project meeting documents, internal research papers. You increasingly could even have old ELNs, legacy ones where all you have left are the PDFs. It's an absolute gold mine. There have been many attempts using SharePoint, using Documentum, using more sophisticated methods to unlock this, but clearly that survey says those have not worked. But this is exactly what large language models are really good at. They can basically take any body of information, typically the internet, and summarize it for you. So if we can take a large language model and train it on all the documents within your informatics system or accessible to your informatics system in the Jarvis paradigm, then you can do exactly the same thing with your internal knowledge. Now, I'm in one of the few companies that doesn't have that huge legacy of documents. So basically what I did is downloaded 10,000 medical papers from the internet and loaded them in to Jarvis like they'd come in from one of our transactional systems. Then I can go in and I can simply say, hey, is there any evidence of COVID infection causing seizures in any age group? Very nice summary. Yes, there are 6 papers of interest in those 10,000, and here's essentially what they're telling me. And again, because this is a chat paradigm, you would expect now to expand the chat. So let's imagine that second paper looked interesting. If I click on that, COVID-19 amongst infants, it's a really big review, looks potentially useful, but it's a very big review. I don't feel like reading the whole thing. See the message Elaine button at the bottom there. Hey, Elaine, please summarize this document. This document is a comprehensive narrative review, et cetera, et cetera. Yeah, looks really good. This is what I was asking. Yes, I had better read this paper. So very quickly with a chat, I've refined down all the way through all my legacy documentation. The third use of large language models is a bit more mundane, but actually very powerful to help scientists get up and running, and also for those of you in research IT, which is we've trained a large language model to be a support assistant. It knows all of our documentation, it knows all of our release notes, it knows our API. So here's an example. This actually comes from the ELN and not Jarvis, but how do I create a plate and add samples? That's all discussed in our documentation, but there's a whole section on making plates. There's a whole section somewhere else on adding samples. Nowhere is there a nice 1 to 5 list. But what the large language model can do very nicely is just assemble that procedure for you, so you don't have to go through the documentation and flick through the indexes and piece it together for yourself. Same thing is true for those of you on the research IT side who then need to take our platform capabilities and integrate them with your landscape. I am not a Python expert. I am not an expert in our API. But even if you were both of those things, I bet you I could make this code snip faster than you could, because I can simply type a prompt. How do I create an experiment called test experiment based on the test template in Python code? Back comes the Python code, really nicely documented, exception handling built in, and even a little summary at the bottom to help me understand what the code snip does. So that's where we stand today with our AI capability. It's in the platform available to our users. The next step then is to give it even more scientific intelligence. So if you think about what does a scientist do? They've assembled a set of data, which they could do in Jarvis. What they typically then do is ask scientific questions. And by that, I mean not just text and numeric-based questions, which large language models are very good at, but they actually ask questions that only a bioinformatics algorithm can answer, or a cheminformatics algorithm can answer, or an image analysis algorithm can answer. Large language models can't do that for themselves, but you can actually give them access to all the tools that a scientist would have. That's how you turn this into a true scientific system. So a very simple example in a video, just to finish up. Imagine I have a sequence loaded into the sequence editor in our software, and I want to do a codon optimization. I don't know how to do that. I'm going to ask Elaine, hey, optimize this sequence. She doesn't natively know how to do that, but she has access to a bioinformatics suite. So she will go off, identify where the sequence is in the software, and has actually come back and done my codon optimization. And just for my reassurance says, I've run the DNA chisel codon optimization. And here's your optimized sequence compared back to your original sequence. So that's just a very brief glimpse into where we're going to go from here with the scientific assistant. And with that, thank you very much, and we'll be happy to answer any questions.
Thank you, Rob, and thank you for your very informative presentation. We will now move over to our question and answer session. First up, we have a question about the LLMs. Which large language model technologies do you use?
So I deliberately didn't answer that question. Obviously, as everybody knows, there's a big range from ChatGPT to Llama, Claude, and all the other ones. We have deliberately set this up so that we're agnostic to which model we use. The really good news for anybody consuming these models at the moment is there's an absolute arms race going on. So, you know, ChatGPT 3.5 came out, and then Llama 2 came out and was better, and then ChatGPT 4 stepped beyond it, and then Claude stepped beyond that. So we've really set it up so that we can actually mix and match. And even in those examples I showed you, depending on when I actually produced them, it could have been using different models behind the scenes.
Okay, and what kind of applications can be linked into Jarvis?
The only thing we really need is an application that has enough of an API that we can query it and essentially say, give me all the information of certain types that's new since the last time I ran this query. So I showed you, and I focused really on ELNs and LIMS, but you can imagine registration systems, you can imagine inventory systems, you can imagine lab execution or method execution systems, you can imagine hooking up your ERP and bringing information in. It's really all the information you need that when a scientist comes in, that's the complete body of information they need about whatever the question they have. It's comprehensive across your company.
Okay, and what kind of scientific questions can you imagine Elaine being able to answer?
The ideal would be if a scientist would ask the question, Elaine would be able to ask it on their behalf. Again, the whole point is to have an assistant that can do for the scientist what they would do themselves, but it takes them time and effort that they could spend somewhere else. So I just showed you a very simple bioinformatics kind of codon optimization. You could absolutely imagine, for example, in medicinal chemistry, you could imagine, here's a molecule, tell me ways in which you think I could make this, or here's a synthetic route. Do you think this route's going to work? Are there better ones? No. All of the kind of things that, you know, a synthetic chemist would go off. Or once we've got activity data, tell me about the SAR patterns in here. What would you suggest the next molecules are? Again, a chemist would go to a cheminformatics algorithm to answer those. So it really is just limited by understanding the kinds of questions a particular scientist in a domain would ask, and then ensuring that we can hook those tools that they would use to answer that into Elaine. And it's quite interesting when you think about the net result, because the net result of this is we can make those tools easier to use than they are to use themselves. Again, I'm no bioinformatician. If I had to do a codon optimization for myself with a bioinformatics code, I would be flailing around for quite a long time to figure it out. But I can just type the prompt and say, hey, do the optimization. I don't even need to know how the third-party software works. You go figure it out for me and just give me the answer.
Do you have, are there any plans to kind of expand this functionality at the moment?
Yes, we are constantly expanding. I mean, this is a huge area of active research. So the three things I showed you with Elaine prompting, searching, the thing I didn't show you because this was a talk about Jarvis and getting data out is Elaine can also actually create information in experiments. So if I were an ELN user, I could say, hey, Elaine, create me an experiment with 8 blood samples and lay them out on a plate, each replicated 10 times. And instead of a lot of clicking and building up an ELN section, a single prompt can have Elaine build up a 3 or 4 section experiment. And then the other really important thing to understand about that is this is about chat. So that's a single prompt, but then you could say, hey, Elaine, add a buffer to every well on the experiment. Hey, Elaine, prepare the liquid handler input file for me. You literally build experiments or build up and refine searches by having a conversation. So yeah, really big area of active research and constantly expanding the capabilities.
Great. Well, thank you very much. That brings us to the end of today's webcast. This content will remain live on Scientific Computing World website at www.scientific-computing.com for the next 12 months. So please spread the word if you have enjoyed this presentation. Of course, please note the live question and answer session will not function if you are listening on demand. Once again, thanks very much to our speaker, Rob Brown, and our sponsors, Sapio Sciences. Thank you all for listening, and goodbye.
Thank you. Bye.